Hello, everybody. Welcome back. This week, we're going to be focusing on each of our major senses, and specifically today and tomorrow, we're going to be looking at vision. Today's more focused on the anatomy of the eye and kind of just walking through step by step how light enters the eye, gets processed, goes up to the brain, yada yada. So, when we talk about what it is that's actually happening first in vision, we know that it's actually light waves are what's entering the eye. Remember we talked about transduction, right? We're taking it in from that sensory mode of like light waves, sound waves, and then turning it into brain language. So, what's first entering our eye are light waves. And let's talk about these waves. So, differences in wavelengths can create differences in the colors they're producing and in how bright they are. So a hue is the color that we're actually seeing. So red, green, blue, whatever. And this is determined by the wavelength, aka how long or short a wave is. So we see here, if you have a short wavelength, meaning they're pretty close together, then that might produce more bluish hues. If you have long spread out wavelength, then that might produce more reddish hues. So hue corresponds to wavelength and kind of how bunched together or far apart and spread apart the waves are. Now, intensity is the brightness of a color. So is it going to be more muted and subdued, or is it going to be very bright? So this is determined by a wave's amplitude, meaning its height. So you see if it's really large amplitude, meaning it's very tall wavelengths, then it's going to produce more bright and intense colors. If we have more smaller amplitude, meaning the wave height is much shorter, then it's going to produce duller, more muted colors. So that's all. Again, the waves are entering our eyes. So here's kind of the overview of this step-by-step process that we're going to spend today going through. And I'll go through this after I go through each step in depth. So first step in our vision process, you're going to have your light waves reflect off of objects. So what's actually happening is when you're looking at something, the light wave bounces off of that and reflects back into your eye. So light waves bouncing off, reflecting in your eye, and it enters first through the cornea. The cornea is just your protective outer layer covering the eye. And this is just there to prevent things like dirt and dust and grime and stuff from getting in there and messing with your eye. So it's going to go through that protective outer layer. All right, so there we got it. Now, what happens is the light's actually going to go through the cornea and now through our pupil. The pupil is just, all you need to know about it, it's just the hole through which light enters our eyeball. And if you've ever been to the eye doctor, they might have done this before where they give you the little medicine to dilate your eyes so you actually can see before you your pupil opening and closing. So that's what happens when you go to the eye doctor and they do that dilation drops is your pupil is opening and closing. And that's the same thing when you're in a dark room. Your pupil is going to expand so it can take in more light. It's going to dilate and take in more light so you can see better. And when you're in a really bright setting, it's super sunny out, whatever, your pupil is going to constrict and close because you don't need as much light to take in because you can see well. So that's just the pupil. It's just the hole through which light enters. It's going to open bigger to let in more light. If it's dark out, it's going to close up a little bit, constrict if it's already bright out and you don't need more light. Now, the iris is just the muscle that is controlling the pupil and opening and closing that. So iris is what's actually the muscle controlling that dilating or constricting of the pupil. And again, back to the eye drop example, the reason why, at least I had all these eye issues last year that I had to deal with, when they did this, it's really painful kind of, and that's why you have to put the sunglasses on and those silly glasses, because when you're dilated during the day, we shouldn't have our pupils that far open during the day because we're taking in way too much light than what we need. And then that's kind of painful because it's too bright. All right. So we got here cornea, pupil, iris. Now let's get into more of the magic happening here in our eye. Okay. So the light waves have officially entered our eye, right? They went through the pupil. So now there's a figure in the eye called the lens. And the lens is going to do the important job of, and think of it like a nice camera lens on like a fancy Canon or whatever camera. It's going to, or DSLR, I think is the name, right? It's going to go kind of like this, and you're going to focus in and kind of, focus and make it more clear and focus those light waves and then project them. So it's an upside down image on the retina. So it kind of flips it upside down and projects it like a little screen projector on the retina. And there's a process that happens in this called accommodation. And this is, again, this actual process of the lens changing the shape and kind of zooming in, zooming out, focusing in on so that image that's projected on the retina is going to be an accurate image. So, right, focusing near or far objects on the retina. It's going to thin or thicken depending on how near or far it needs to be, how far the object is. So you can think of the lens as kind of like that DSLR camera zooming in and out. And accommodation is that process of zooming in and out to focus and project it, flip it upside down on the retina. All right, so now the magic happens in the retina. This is the most important part because this is where transduction occurs. So we're going to take big notes on this one. So we said lens, boom, flipped upside down, focused it in, and now the image is projected on the retina. The retina contains our photoreceptor cells, which are called rods and cones. And it's really important to note that it is with the rods and cones in the retina that is where transduction takes place and starts. So, image is projected in the retina. The rods and cones are going to help process that and begin the transduction process of changing it from light waves into neural impulses, the brain language, which the brain can understand, so it can be sent up to the brain for processing. So the rods process kind of your nighttime peripheral vision. So it's more of like black, white, and gray is what it's processing. So it's more for nighttime or peripheral vision. Cones, it's easy to remember. Think CC, cones process colors. So this is more for your daylight like images that you're seeing. And they also help more with fine details. And these are located in an area in the retina called the fovea. So again, really important to note that it is within the retina, specifically with those photoreceptors, rods and cones that are processing those colors for you, where transduction occurs, and we're changing it from light waves now to the brain language of neural impulse that the brain understands, so it can be sent from the retina and exit the eye and now be sent up to the brain for processing, up in the occipital lobe and all that jazz. So take big notes here because you're going to need to know that this is where transduction happens. All right, so now we've had transduction occur. And we're going to see now we're no longer talking light waves. We are talking neural impulses. And these are going to go through two different types of cells called the bipolar and the ganglion cells. So now we're talking neural impulse, no longer light waves. They're first going to travel through bipolar cells. Don't really need to know anything else about these except that they're just traveling through them. But then there's the ganglion cells. And these are important cells to note because they make up the optic nerve. And the optic nerve is kind of what takes the neural impulse now from, kitty. The ganglion cells is what takes the neural impulse away from the eye and up to the brain. So it's kind of the exit door of the eye going up to the brain. So ganglion cells are important to note because they're what make up the optic nerve. Another thing that's important to note about the optic nerve is it actually creates our blind spot. So you can see here in this picture, the optic nerve, it's where the blind spot is, where we just, you can't see. It's like a blind spot here that's kind of blocking it. So where this happens is where, just noting that the optic nerve is where the blind spot in the eye is located. So again, now we got neural impulses moving through these cells, go out the optic nerve made up of ganglion cells, and now they have exited the eye, so we are moving and grooving. Okay, my little Frankie cat messed up my slides. Okay, so we exit the optic nerve, and like with any sensation, it's going to be sent up, unless it's smell, it's going to be sent up to the thalamus to be organized and sorted out in the little mail room of the thalamus before it goes to its appropriate part of the brain or the lobe for final processing. So now we're here, boom, made it through the cells, exited the eye, and now we're going to send it up from the eye to the thalamus to get all sorted out. So, right, we're going to be in the thalamus, it's going to be organized and then relayed and shipped to its final destination where it's actually processed, because we know that the thalamus doesn't technically officially process it, it's just kind of sorting it out. So thalamus can be, hey, yo, this is visual, send it on up to the occipital lobe. Bam, there we are. Yep. And we're now in the occipital lobe, which we know is mainly for processing vision. So specifically, we should note that it is the visual cortex, within the occipital lobe, which is what processes this. So occipital lobe, we know that there's kind of more things within these lobes than just the overarching lobe. So it is specifically the visual cortex in the occipital lobe that processes this information and goes through the process of actually recognition and all these things that we talked about before. So there are specifically things in the visual cortex called feature detectors, and these are what's going to help us recognize what we're looking at and kind of put a label and name to the face, right? So it's going to help us identify specific features of a stimulus, thinking like shapes, edges, lines, angles, right? Figure out, again, helping us figure out what it is we're looking at. So now we've made it. We have one last thing called parallel processing, which is just a thing to note that is kind of a cool thing of how we process things. We're not just doing it one step at a time. We kind of have a bunch of processing going on. My kitty's making a face. We have a bunch of processing going on at once in which we're not just going through, okay, let's look at the form, let's look at the depth, let's look at the colors. We're processing all these things at once. So when it means parallel, right, you have kind of two tracks going on at once. It just means that all these things are occurring at one point in time, that we're processing multiple things, looking at the color, the form, the angles, and all this stuff of what we're viewing all at once, because our brains are very speedy things. And then finally, recognition occurs. Yay! We go through parallel processing, we're doing all the stuff, and we are going to connect it to our stored information and memories. So now we can figure out what it actually is we're looking at and go through the perception part of sensation and perception. So there we go. And we're just going to end by talking about two theories of how we process color. So there's the trichromatic and the opponent processing theory of color. And we're going to look at how both of these would explain color blindness. So trichromatic theory, we know that tri means three. So trichromatic theory says that there's three types of cones. And remember I said that there's the two kind of color processors, which are the cones and the rods, or two photoreceptors, which are the cones and the rods. Cones specifically process colors. So cones, they're saying in the trichromatic theory that there's three types of cones that are contained in the retina. And they said there's specifically one that detects red, one that detects greens, and one that detects blues. And from there, they basically, if you understand art, this might make a little more sense to you in understanding how colors blend together. But they're saying that cones are kind of activated in different combos, and then that's what producing other colors beyond red, green, and blue. Just like if you have a little artist palette, right? You don't need to have 200 different paints because you can just blend your, main colors together to create other colors. So that's what they're saying here is you're kind of activating greens and reds, and that'll produce a color. Again, I'm not bad at knowing all my mixing colors. My mom would be ashamed. She's an art teacher. But for example, red and green together, if they're both stimulated, would create yellow. So what they're saying as a cause for color blindness would be that you're lacking a functioning cone. lack of functioning cone, right? So maybe you're missing your red cone, and that's going to create color blindness. So trichromatic theory, again, just saying that we produce colors and that we have 3 types of cones, and they're kind of blending together, being stimulated at different times to create that sensation of a different color. Opponent processing theory is different in that my cat is eating my charger. No. Opponent processing theory says that we have pairs of color receptors. So we have like a red and green pair, a blue and yellow pair, and a black and white pair. And what happens is if you have one stimulated in a pair, so you have your red being stimulated, if you're looking at the red too long, you exhaust that receptor, and then you're going to see the opponent in the pair. So, right, it's like red, they're kind of like opposing opponent pairs. So if you're looking really hard at red, let's give that example, you're going to eventually exhaust that color receptor. And when you stop looking at it and look at kind of a blank white screen, you'd see green as the after image. So again, this is called sensory fatigue of 1 is going to cause the opponent pair to fire. So again, fatigue of green might cause red to fire and vice versa. So how this one explains color blindness, it does it in kind of a nice way, saying that if you're missing a pair, you can't see those hues. And this makes sense. You often hear like a red-green colorblind type thing. So that's kind of like something that might make sense in explaining color blindness. So you can practice this after image. So if you stare at this white dot for like 30 seconds and then go look at this blank page, you're likely to see the after image of the opponent pairs. So if we're seeing yellow and green, it would follow that we're going to, once we look at the blank white one, we exhausted our yellow and green, so we're going to see opposing pairs. So we've exhausted green, we're going to see red, and we've exhausted yellow, we're going to see blue. So that's how opponent processing explains colors. So there you have it. And that is some color theory for you. So you guys are going to create your own little Play-Doh eye and run on through that whole process and practice that. All right, have